Welcome to this new topic, growth and development. So we are nearing the end of grade 11 syllabus. When we get done with this topic, we're going to remain with two topics. Once we deal with those two topics, then we'll be in for the final part, which is grade 12. So this is going to be delivered into three separate videos. In this video, we're going to take a look at introduction to growth and development. Then after that, we're going to take a look at growth in plants. Then we're going to continue growth in plants by taking a look at germination. Then we're going to finish this topic by taking a look at growth in animals. Let's get started. So growth is defined as a permanent increase in size, mass, number of cells, and complexity of a living organism. So we're going to look at some of these growth patterns by looking at life cycles. So a life cycle is a sequence of stages that an organism passes through during its development from the embryonic stage to maturity. So an organism, of course, starts as an embryo. So we're going to study how it develops from the embryo till it becomes a mature organism in what are called life cycle. The reason why it's a cycle is because when it's mature, it's going to create young ones in form of embryo. Then the cycle will continue like that. So we're going to start with growth in plants. So plants undergo two types of growth, namely primary growth and secondary growth. Primary growth is the increase in length of the shoots and roots. So a seed has an embryo made of a smaller shoot and a smaller root. A root is what is going to develop to become roots for that plant, and the shoot is what is going to sprout from the ground, and it will be a plant that we're going to be able to see, which is going to be above the ground. Then the other type of growth, which is secondary growth, is just the increase in the width or girth of shoots and roots. So first it has to the shoot and the root have to develop. When that happens, then the plant is going to increase in the width. So that is secondary growth when it is increasing in the width or girth. So what's the purpose of primary growth? The purpose of primary growth is to enable the roots to penetrate the ground and the shoots to grow towards sunlight. Once the shoots are able to access sunlight, then other things are going to become easy and the plant will be ready for secondary growth. And once the roots also penetrate into the ground such that they can get water deep in the ground, and of course they can support the plant mechanically. They can offer mechanical support to the upper parts of the plant by holding the plant in the ground. Then the plant will be ready for secondary growth. So plant growth basically involves three stages. Okay, remember the types of growth that a plant undergoes: primary and secondary. So each of these has three stages. So the first is called cell division, where a cell divides itself so that they are many. So you can have one cell, it divides, they become two. Those two divide as they become four. So cell division is just the increase in the number of cells from one single cell or a few cells to more cells. That's what cell division is. So one cell can divide itself, they become two. Two cells can divide themselves, they become four, just like that. The second is what is called cell vacuolation or cell elongation. So this has to do with the vacuole of a plant cell. Okay, so there is an activity that happens to the vacuole that results in it increasing in length or elongating. So we're going to look at that. The third is cell specialization or cell differentiation. So in cell specialization, remember these cells first start as a single cell, then they have to keep on dividing. So at some point now, each of the cells or some of the cells have to know what their purpose is. What are they going to be doing? Are they going to be xylem cells? Are they going to be phloem cells? Are they going to be palisade cells? So that's cell specialization or differentiation. So we're going to take a look at each of these stages. So start with cell division. So this is the process by which new cells, which are called daughter cells, are formed from cells that are already existing, called parent cells, like I explained. The type of cell division involved in growth is called mitosis. Okay, so there are two types of cell division. There's mitosis and meiosis. So for this growth, or for growth, or in this part, the type of cell division that happens is mitosis. So we're going to learn about mitosis and meiosis when we look at genetics. For now, we are just going to have this basic idea of what mitosis is. So mitosis is a type of cell division where one parent cell produces two daughter cells that are genetically identical to the parent cell. So genetically identical, it means they have the same types of genes as the parents. Okay, so what are genes? Genes are units of inheritance. They are things that makes you look like your parents. Okay, so in mitosis, the daughter cells are going to be exactly like their parents. So regions of active cell division are known as meristems or meristematic tissues. So regions of active cell divisions are known as meristems or meristematic tissues. So here you have a plant where cell division is happening. Those parts are called meristems or meristematic tissues. So there are two types of meristematic tissues in plants. The first are called apical meristems. The second are called cambial meristems, or simply cambium. This cambium, this is not the first time you have come across this word. Remember, when we were talking about transport in plants, we talked about the cambium. So that cambium is there to facilitate cell division. Okay. Then the apical meristem. Sorry, there are some kids who are making noise outside. So the apical meristem, the word apical comes from apex. The apex is like the tip. So this is the type of this is the part of the plant which is found on the roots or shoots at the end or the tips of the shoots and roots. Hence apical or apex, which is just the tips. So apical meristems occur at the tips of shoots and roots and are responsible for primary growth, like I said. Well, cambium are responsible for secondary growth. So the apical meristems make sure that the primary growth occurs successfully, while cambium makes sure that secondary growth occurs successfully. So that's all about the first part, which is cell division. Remember, the cells that are made from cell division are genetically identical. They are not different from each other or from their parents. The next stage is cell elongation or cell vacuolation. So this is a process by which cells grow bigger and develop their vacuoles by absorbing a lot of water. Remember, water and mineral salts are stored in the vacuole, so they are going to be absorbing a lot of water, and because of that they are going to elongate, or they are going to increase in length or size. Okay, so that's the second stage. One thing to note is, after being vacuolated, all cells still look identical. There's nothing, there are no new cells, they all look the same. Okay, so just like in cell division we have the same types of cells, in cell elongation we still have the same type of cells. The last stage is cell differentiation or cell specialization. So this is a process by which cells become suited for specific functions by developing specific shapes and undergoing specific chemical changes in their cytoplasms. So here, that's when we get different types of cells now. the xylem cells, the phloem cells, the spongy cells, the palisade cells, different types of cells. Now, one thing to note, or one thing that makes cell differentiation different from cell elongation and the cell division is the cells are going to be different, unlike in cell elongation and the cell division where all the cells are the same. In cell differentiation, they are going to be different. So now we're going to take a look at what happens at the apices or tips of roots and shoots. So there are three regions of growth at the apices of shoots and roots in plants. So whenever you get a plant, the tip of the plant, the root and the shoot, it has three parts, each part undergoing a different type of growth. That is cell division, cell elongation, and cell specialization. Okay, so let's take a look at oh, by the way, the regions, like I've said, is regions of cell division, elongation, ovacculation, and cell differentiation or specialization. So if we take a look at a shoot, this is an example of a shoot. Okay, so a shoot is the part, of course, that sprouts from the ground. So if you look at this shoot, the upper part is this, it's going upwards, so the newer cells are on top while the old cells are here on the bottom. So these older cells have already gone two types of cell division, two types of growth, two stages of growth, that is cell division and the cell elongation. Now they are undergoing cell specialization. They are becoming vascular systems. They are also developing into other types of tissues. Into other types of cells. Okay, so these cells have already gone specialization. Okay, so the next part is this, which is the region of cell elongation. So this is the part where the cells are increasing in size or elongating. So they are the ones that are making this shoot to get taller. Okay, so in the region of cell elongation, the cells have already gone cell division. They are now on cell elongation. When they are done with cell elongation, they are going also to become regions of cell specialization. But at this specific time, at this snapshot, when we're studying this shoot, this part is undergoing cell elongation. Then on top here, there are new cells. These are the cells that are being made or that are being generated through cell division. So these are just baby cells. With time, they are going to undergo cell elongation and later cell specialization. But at this specific time, they are undergoing cell division. Okay, so this is how to comprehend a shoot. The bottom, these are old cells, so they've already gone cell specialization. They've already gone cell division, cell elongation. They are now undergoing cell specialization. These are a bit older than the ones on top, so they have already gone cell division. They are now undergoing cell elongation, and these ones are undergoing cell division. So the shoot is opposite in the sense that the shoot is going downwards. So expect the part on top. This is a root, sorry. So for the root, it is going downwards. So it's opposite to the shoot. So for the root, this part has older cells. So these cells have already gone two stages, cell division, cell elongation, and they're undergoing differentiation. So this is the region of cell differentiation. The second is this part where the cells have already undergone cell division. Now they are doing cell elongation. And the last part is this, which is the region of cell division. Same analysis, same thinking as here. It's just that in the roots it's going downwards, in the shoot it's going upwards. Next, we take a look at germination. So germination is the process by which seedlings develop from seed embryos. Remember, all growth starts with embryo and then with a mature stage. So germination now is the process by which seedlings develop from seed embryos. So we're going to start germination in the two types of seeds. monocot seeds and dicot seeds. Monocot is short for monocotyledonous seed, and dicot is short for dicotyledonous seeds. So we're going to take a look at the monocot seed, an example being a maize seed, and also going to take a look at a dicot seed, an example being a bean seed. So on the part of the monocot, the embryo is this part. Okay, so if we start the embryo of the monocot, it is made up of two most important parts, which are the plum here, plum, and on the bottom there is a radicle. A plume is a part that develops into a shoot, while a radicle is a part that develops into a root. Additionally, the plume and the radicle have a protective covering which help when a plant is when the shoot is trying to break the ground and go up, and when the root is trying to go deep into the ground. So this part here protects the plume, and this part here protects the radicle. So this part on the radicle is called a coleorhiza, well the part on the plume is called a coleoptile. Okay, so this is the embryo for a monocot. For a dicot, we don't have everything we have on the monocot. For the dicot, we have a plume here and a radicle here. Just like in the monocot, the plume develops into a shoot, a radicle develops into a root. Okay, this makes the embryo for the dicot. Other than that, we have other parts. On the monocot, we have a cotyledon. This is the cotyledon here, and on the dicot, we have this big cotyledon. So these two cotyledons have different functions. In the dicot, it is used to store food. Okay, well in the monocot, it is used to digest some substances on behalf of the embryo or for the embryo, and also it has other functions. Okay, but in the dicot, this big cotyledon is used as a food storage. Okay, so another part we can see that is present in one but absent in the other is this opening here, the micropyle. Okay, where some substances pass through. Well, here we have got the endosperm. Okay, so seeds that have the endosperm are called endospermic seeds. Well, those ones which don't have the endosperm are called they are called non-endospermic seeds. So for a monocot, the endosperm is used as a food storage instead of the cotyledon. Well, this one doesn't have the endosperm, therefore it stores its food in the cotyledon. Okay, then another important part which is present in both is the tester. You can see the tester here, and here also there is a tester. So the tester has one job, and that is to protect the seed, the internal parts of the seed, okay, from external damage, external shocks, and the likes. So these are the main parts of the monocot and the dicot. So a summary of what I've just explained, we take a look at each part and their functions. So a tester is responsible for protecting the seed against physical damage and also infection, which I did not mention, infection. So it makes sure that germs or bacteria don't enter the seed. It is also responsible for a process called dominancy in certain seeds, where it prevents a process called imbibition, which is simply the entry of water into the seeds. Okay, so imbibition is just the entry of water into the seeds. We're going to start dormance later. The plume. So this is the embryonic shoot. It develops into a shoot after germination. The radicle is the embryonic root. It develops into roots after germination. So the plume and radicle together makes up the embryo of the seed. So the region of the embryo next to the plume is called the epicotyle, while the region next to the radicle is called the hypocotyle. So just that region next to the plume is the epicotyle, and the region just next to the radicle is called the hypocotyl. The next is the cotyledon. So these store food and enzymes in most dicot seeds. Dicot, not monocot. In most monocot seeds, food is stored in another tissue known as the endosperm, like I explained. So seeds which have the endosperm are called endospermic seeds, while those without the endosperm are called non-endospermic seeds. The main form of food stored in seeds includes starch, oils, and proteins. Starch, oils, and proteins. Some of these, especially oils and starch, we discussed that their main storage, their main form in which food is stored in plants. So in some monocot seeds, such as maize, the plume is protected by a sheath called the coleoptile, while the radicle is protected by a sheath called the coleorhiza. So all this information was shared. So these prevent damage during germination. So the shoot, as it is trying to sprout, the coleoptile is going to protect it to make sure that it is not damaged, and also the root, when it's about to dig deep into the ground, the coleorhiza is going to protect it. So this marks the end of part 1 of growth and development. Hope you have enjoyed. Happy solving.